right, so we've determined the natural selection, first off, can't make anything. You can only select and eliminate. And even then, its elimination process is a bit weak. I think one analogy has been put forward. It's like the princess and the pea. You really don't have that much sensitivity to natural selection as you would need for it to serve the purpose that evolutionists want it to serve. But it comes back to, okay, if natural selection is not the one that's generating the traits, it's only selecting through the traits, then where do you generate the new traits? How do you get the wings on the car? How do you get the legs on the fish? And that's through a process of mutation. Mutations then are seen as the main mechanism for driving the stepping or the climbing up the mountain. Okay, so very quickly, we're going to define mutation as any change to the nucleotide sequence of DNA. If you remember your biology, DNA is 4 nucleotides, adenine, thymine, guanine, cytosine. They're abbreviated as, of course, TACG. When you change one of those nucleotides and that change becomes fixed, in other words, it becomes permanent, it's not repaired by the repair system, then that becomes a mutation. Now, the consequence of that mutation can be a lot. It can have very little consequence. It can have consequences that would potentially make the organism maybe more fit, less fit, neutral. And that's what a lot of this study in mutations is about. Well, what effect did the mutation have? What did it do to the organism that altered it? Did it change its color? Did it make it faster? Did it make it slower? Did it make it taller? Did it make it shorter? What did the mutation do? All right, so when we go back to universal common descent, then the idea is that through the filter of natural selection, the mutations have provided us, or beneficial mutations have provided us then the mechanism to create the changes so we get this kind of diversity, this kind of transformation occurring. So the fish becomes some type of land creature, which becomes a reptile, which becomes a bird, which becomes a mammal, which then ultimately becomes a primate and then becomes a human. So we have mutations, specifically beneficial mutations, in other words, mutations that are supposed to be good for us, that are occurring all along the history of life, changing us ever upward in this evolutionary panorama, if you will, to give us the life or the diversity of biology we see today. So that is the power that's being invested into beneficial mutations. Now, I need to introduce a couple terms very quickly. In biology, when we talk about a phenotype, we're talking about the physical traits, the physical characteristics, your hair color, your eye color, your height, whether you have 5 fingers, whether you have 2 arms, that's your phenotype. The genotype then is the genetic system behind that phenotype. Now there's a relationship of the two, but obviously we're still learning some of the relationship. We're finding out that there's very interesting situations of certain genotypes giving certain phenotypes and not giving other phenotypes. So that's still part of where the field of genetics is at. But the genotype refers to the genetic system in the organism. The phenotype refers to the physical characteristics of the organism. And the reason that's important to understand is because I'm going to challenge that all too often in the literature, in the scientific literature, when they talk about beneficial mutations, they are almost exclusively identifying and categorizing them by their phenotype, with little or no regard to their actual genotype. In other words, okay, you got this phenotype, you got this change that occurred in the organism, but what happened at the DNA level to bring about that change? And let's look at some examples to understand what I'm getting at. Okay, malaria resistance. Obviously, we've heard of sickle cell anemia as a way of resisting malaria. Of course, that's a pretty drastic way to resist malaria because you may not die of malaria, you die of sickle cell anemia instead. So I'm going to offer the evolutionist community a much better mutation, and that is the Duffy mutation. You have on your red blood cells a protein called the Duffy antigen or the Duffy protein. And the parasite that causes malaria will bind to that Duffy protein, and that's what helps it get into the red blood cell to give you malaria. But if you have a mutation that eliminates that Duffy protein, so the red blood cell does not carry the Duffy protein, the parasite does not enter in the red blood cell very well, and you are resistant to malaria. So is that a beneficial mutation? If you're in an area where there's a lot of malaria, it absolutely is beneficial, very much so. But look at what's happening at the genotype, not the phenotype, but at the genotype. At the genotype, you've had a mutation that is inactivated an already pre-existing gene. Now, any mutation that inactivates a pre-existing system does not give you an explanation where that system came from to begin with. So this may drive diversity, but it does not drive climbing up the mountain. You don't go up the mountain by eliminating stuff. That's stepping down on the mountain. That's going backwards. So if we want to go up the mountain, we got to have mutations that are going to go upward. We're going to have mutations that are going to explain how we got this to begin with. Okay, let's do another. DDT resistance in insects. I see this in textbooks all the time. Evolution of insects. They became DDT resistant. Well, there seems to be a number of very specific ways that insects develop DDT resistance, but in general, a mutation is introduced into a regulatory component of the CYP6G1 gene. That gene is responsible for making a protein called P450. And the mutation creates an excess amount of P450. It doesn't eliminate the making of the P450, it makes an excess amount of P450, which is what contributes to the insect's DDT resistance. All right, let's look at this again. At the phenotype level, beneficial, the insect resistant DDT, it still lives, it's evolved. Okay, but at the genotype level, what has happened? A mutation has wiped out the regulatory control of this gene so that it is now overexpressing, is making too much of the P450. So this mutation cannot explain not only where the P450 gene came from, but it can't explain where the regulation of this P450 gene came from because the mutation knocks that out. So again, we're knocking out a pre-existing system. It's giving us a beneficial phenotype, but the genotype is we're eliminating pre-existing systems. Another example, HIV infection. If you are exposed to the HIV virus, it binds to a protein called the CCR5 protein, which is on one of your immune T cells, one of the cells part of your immune system. Okay, and that virus then, once bound to that CCR5 protein, will be able to infect that cell, and potentially you would potentially be able to get AIDS from that. All right, now there is a small population of people that have a mutation in the gene that makes that CCR5 protein, and they no longer have it on their T cell, and therefore the HIV does not bind or infect their T cell, and so they are in effect resistant to HIV. Now, beneficial phenotype, absolutely. If you're exposed to HIV, it's nice to be resistant. But let's go back to the genotype again. What's happening at the genetic level? The genetic level, we have knocked out a pre-existing protein. So this mutation doesn't explain where the protein came from to begin with. It only explains how we knocked it out. So again, that's not stepping up the mountain, that's going backwards. That's stepping back down off the mountain. And human lactose tolerance. Most children pre-puberty make a enzyme called lactase, and they're able to digest the lactose of milk. But as they pass through puberty and become an adult, there is a regulatory system that kicks in that shuts the lactase gene down, and they no longer make the lactase enzyme. Therefore, they cannot digest the lactose in milk, and they would often be what we'd say have a milk allergy. Now, this is actually most people, or the majority of people in the world, are lactose intolerant. They do not make that lactase gene after post-puberty. But there are some of us, especially those of us that have more of a Northern European ancestry, or at least some of our ancestors were Northern European. Some of us have a mutation in the MCM6 gene, which is involved in regulating the lactase gene. And that mutation keeps then it from shutting down the lactase gene, and you continue to make the lactase enzyme through your adult life. Now, is this beneficial? Well, I'm glad I have this mutation because I like milk. and I like drinking milk, and I'm glad as an adult I can drink milk. So yes, it's beneficial if you want to drink milk, but once again, let's look at it. Genotype wise, it's a mutation that knocked out a regulatory system. So this mutation does not explain how you climb Mount Improbable. It only explains how you step backward off Mount Improbable. In other words, losing systems that are already there to begin with. Antibiotic resistance. It's amazing to me that as creationists, we have to keep talking about this, but the evolutionists, they persist. They keep it in their textbooks. They keep referring to it all the time as examples of evolution in action. Well, again, it's an evolution in action only if you're defining evolution simply as change. But if you're going to the deeper component of saying, but evolution, ultimately you're looking at is universal common descent, how does antibiotic resistance explain that? And the answer is, of course, it doesn't, because it's not providing that kind of genetic mechanism. Antibiotic resistance says that you have a population of bacteria and they're exposed to an antibiotic. Those members of that population that have a resistance in some manner by some mechanism will survive this antibiotic. So certainly for the bacterial population, or the cells that survive anyway, it's very beneficial mutation. They've lived through the antibiotic to live to fight another day, if you will. But let's look at what is a common cause or a common reason for antibiotic resistance. Okay, loss of enzyme activity, halting of cell division, loss of regulatory protein, reduced formation of a poron or regulatory protein, loss of a poron, loss of reduced, loss, reduced, loss, reduced, loss. Do we see a trend here? See, the bacteria are becoming resistant to the antibiotic by essentially eliminating some system that either allows the antibiotic to get into the cell or allows the antibiotic to interfere with cellular systems in the cell. So by eliminating that, then the antibiotic won't have an effect on the bacterial cell. And as such, they can survive the presence of the antibiotic and live, as I say, to fight another day. It's a little bit analogous to your foot is caught in a bear trap and here comes the bear. What are you going to do? Well, see, you may have to decide you got to chop off your foot, although that still may not get you very far, but you decide you have to chop off your foot. The advantage bacteria have is they can chop off their foot but grow it back. You can't grow your foot back, but they can. Once they lose the system, they survive the antibiotic onslaught, but now the antibiotic is removed from their environment. They then go back to actively making that protein, actively making that transport system, actively making that poron. So they have that advantage that you don't have, and that's why they can respond very quickly to very harsh environmental conditions. But once again, let's put it in perspective. What is happening that's giving them the ability, mutation-wise, to be resistant to the antibiotic? It is a mutation that is eliminating pre-existing systems. It is in no way giving the antibiotic some new characteristic, some new gene, some new genetic event that it did not previously have. We're eliminating pre-existing systems. So we're coming back down off the mountain. We're not climbing up the mountain. Okay? The long-term evolution experiment, I would assume many of you have heard of this. This is an experiment that was done at Michigan State University by a professor named Linsky and his graduate students and such. And it's a very good experiment. I love the experiment. I wish I would have thought of it. It's a great experiment for microbiologists, but I would argue that that's all it's really for is microbiologists. But he took a strain of E. coli and he cultured it day after day, week after week, year after year for over 65,000 generations. And one of the key things you have to recognize in this experiment is the media and the temperature were always the same. In other words, the E. coli never saw anything different. For years and years and years, it was the same thing day after day after day. What they found was, yes, they began to get spontaneous mutants that have formed in the E. coli. Some of them were, for example, the E. coli started knocking out DNA it didn't use to shorten the chromosome so it could replicate faster. Okay, but that doesn't explain where the portions of the chromosome that got knocked out came from. It just means that the E. coli found that, hey, if I have a mutation that inadvertently deleted part of my chromosome, that's actually a benefit because I can now replicate faster. Other mutations eliminated various metabolic pathways and such that the E. coli wasn't using. So by and large, all the mutations they found were mutations that Linsky identified as what he called antagonistic pleiotropy. In other words, a trade-off, a sacrifice of a particular existing system that's not essential in a specific environment if it helps increase the adaptation to that specific environment. I don't need this DNA in this environment, so I knock it out, and lo and behold, it actually serves an advantage to me to get rid of it. And so what they saw, for the most part, in all of their mutants was, with just a couple of exceptions, is a trade-off where it would eliminate systems that it didn't need, it didn't use, which wound up allowing it to grow faster, it wound up allowing it to adapt better to the particular environment. So again, if we look at genotype versus phenotype, yeah, we have this phenotype where it's growing faster. This mutant now grows faster than its original parent does, but let's look at the genotype. It grows faster because it got rid of a lot of stuff. It threw things overboard that it didn't need in that specific environment. Now, the moment you took any of these mutants out of that specific environment and put them into a different environment, obviously they didn't do very well. But of course, that wasn't the point of the experiment, but the way that they're interpreting the experiment, we always have to be reminded of that this is a very environment-specific mutation that occurred, a very environment-specific condition, and you change the environment and suddenly it's no good to you anymore.